Neale. He received his Ph.D. from the University of London, and most of his career he's been at Virginia Commonwealth University. I first learned about his work about 20 years ago when I started to work on a paper that I thought I discovered this major flaw in how structural equation modeling programs implemented standard errors and tested significance. So I was just about ready to wrap up the manuscript and send it for publication when I found a paper that Michael wrote, already published, that did exactly the same thing. Not only did he point out the same error, but he also wrote a software package to fix the problem and do structural equation modeling correctly. So now I've been able to interact with Michael sort of off and on at conferences and so on. I've followed his work ever since that point when I learned about him. So it's a great pleasure to introduce Michael Neale.
Rich, thank you so much for that nice introduction and for inviting me here. I've really had a blast today meeting with people and lots of interesting discussions. So although I'm probably known a lot for twin work and genetics, I'm going to talk mostly about measurement because twin work, like anything else, isn't really any different. That garbage in, garbage out still rules, and that's the case with measurement, that you've got to make sure your measures are not garbage in the first place. So here's the traditional table of contents slide. I want to talk a bit about measurement invariance, applications to National Survey on Drug Use in Households data, talk a little bit about mixture distribution models, some developmental models, and to some extent trying to focus not just on the modeling but kind of what you can do in practice with the results of modeling. How can you get hold of improved measures? And finally, if there's time, I'll give a little bit of a nod to the twin work and talk about direction of causation between how big your brain is and how much you smoke. So, and I thought, since I probably won't get through all of this in the time, I'll give the acknowledgements now. Great deal of work by my colleagues at VIPBG and the OpenMX team, the folks who've made the software package Rich mentioned, took it from a standalone Fortran program into an R package, which we're on the point of releasing to CRAN, having got rid of or at least incorporated other optimizers apart from the proprietary one we had been using. And NIDA and NIMH have underwritten a lot of this work, and I'm very grateful to them and to the great colleagues and students that their grant funds have supported and that I've had the fortune of working with. So first up, the measurement invariance issue. And quite a lot of the history of measurement invariance owes to colleagues of Rich and myself at the Society for Multivariate Experimental Psychology. Starting, I think, especially with Bill Meredith. Sadly, two of these are the late, so that's the late Bill Meredith, the late Roger Millsap. Fortunately, Keith Wideman, Emilio Ferreira still alive and kicking. But very much, I think, the discipline, and this last paper is, funnily enough, not from a SMEPI, but I found a very useful low-level introduction to measurement invariance conceptually. And I think one of the reasons that measurement invariance isn't more widely used, it should be pretty much ubiquitous for psychological and behavioral measures, especially the stuff that begins at the binary item level. But by and large, we should be much more widespread in the application, and one of the reasons it's not very widespread is that it's a pain in the ass. Basically, it's lots and lots of model fitting and difficult to get a hold of it, and it comes along with its own little language of configural invariance and structure and so on, and it gets a bit confusing. So to try and deconstruct it, and I won't be using those labels because I don't personally find them terribly useful, but just to concentrate on the parameters of the model that might be influenced by external variables. So here's our good friend, the latent factor model. This is the guy that we want to know about. This is our construct of depression or substance use liability or whatever. There's, I'm obsessional about drawing loop variances on these diagrams, having worked a bit with Jack McArdle and Steve Boker. These loops make the diagram mathematically complete. You could fit this model directly from the diagram because it is complete in describing what the variance of this is. Factor loadings here, residual variances here. The embellishment, this one especially due to Jack McArdle, is the addition of means in a covariant structure model. So whilst the factor bit describes the covariances, the triangle bit here, as if it was like a measured variable that was 1 on everybody, that's one way that you can implement it. We're looking at the regressions of that, and so it gives you a grand mean for each of the items. That by itself, especially in the 3 variable case, isn't particularly edifying since it's a saturated model. But it gets a bit more interesting if we admit that the factor mean is the thing of interest. What we want to know when we compare populations, when we're looking for interactions, experimental effects and so on, has this factor mean changed? And similarly, maybe the factor variance has changed too. These are the quantities that we're after telling differences about between people. And the rider is that when you use a sum score or a factor score, you might get it wrong. You might get completely the wrong answer. You might miss a difference that's there, i.e. false negative. You might think there's a difference when there actually isn't one, false positive. So all sorts, type 1, type 2 errors can occur in inferences about the latent factor if there is measurement non-invariance. Some other components of the model might wind up looking like mean differences between groups. We don't want that. We've got to go after testing all these different components of the model. And so that's factor variances, factor means, factor loadings, residual variances, and item-specific means. You can't do all of the above at the same time. You have to pick and choose a little bit, but when you've got multiple groups or even continuous moderating variables, you can get after these things simultaneously. And that's what I want to talk about in an application to the NSDR data set. So everyone with me for now? Yeah? This, I hope, is not too alien. Okay. So SAMHSA actually collect loads and loads of data, really good stuff. And they do face-to-face interviews with some 50,000 people every couple of years or so, and look at particularly rates of use of various different substances. And also they do it not just by, like any normal person would do with a web survey. They do it with face-to-face interviews and, in fact, sort of computer audio assisted interviews with people. So they're sort of sitting there with the person in the room whilst they're interacting with a computer. So slightly less chance of garbage in than a web survey, perhaps. The marijuana section will basically enable DSM diagnoses of substance abuse and or dependence. And there are various, this would be an example of what an item looks like. Items for abuse are things like causing you to have serious problems at home or at work, or say driving or something like that. So these would be examples of abuse items. Dependence items are more about sort of not being able to cut down, spending a lot of time planning how you're going to get hold of it and things like that. In most studies, abuse and dependence items actually wind up loading on the same factor. There's some dispute, and I'm sure if you get the sample size big enough, you can get some distinction into the abuse and dependence. But I think it was John Jinks who said the number of factors is proportional to the industry of the investigator, meaning that you've got bigger sample sizes, you'll find more factors. So in order, hopefully, to facilitate work with, partly with OpenMX, because OpenMX is a bare-bones software program where you can manipulate matrices and algebras in order to specify models, and that's to make it maximally flexible. But the beauty of R is that once you've got base bones like that, you can build other structures, other functions on top, make it simpler to apply these analyses. So I wrote this little non-invariance function, and you throw it some data, and you throw it which variables are the ones you want to analyze, which are the moderator variables, do I want age, sex, something else, to be possibly changing factor means, race. I generally trust exogenous variables much more than variables that might be correlated with the outcomes for reasons other than being exogenous, things that are consequences of the outcomes. There is treacherous territory over there. You want to be careful about moderators that correlate for reasons other than being exogenous. Age and sex, generally they're not caused by your depression. Then there's just a list of those 5 components of the models. There's a number of factors that you want in the model, but just a list of the 5 parts of the model that could be different between groups or as a function of a continuous variable. So measurement invariance you can work by multiple groups, but you can also work it with a continuous exogenous variable such as age. So here's an example use. Here would be a list of variables put into an object. Here's a list of the moderators. And then I'll just ask it to build a one-factor model. This would be a null model with no invariance, with complete invariance everywhere. The double negative of invariance is magnificent and will trip everyone up at some stage. That's why differential item functioning is a better name for it, but I boobed and didn't use it here. So here's a model where we're going to fit it with testing for factor mean differences as a function of the moderators, just testing for factor mean differences in sex, but not with respect to age. Okay, you didn't come here to get a coding lesson, I guess. So here are 4 models. Here's a base factor model. Here's one that says the means are a function of the moderators. Here's one that says the variances are. And here's one that say that both these things are. And those are all what we would think of as kosher group differences. The other type of difference might be that there's a difference in factor loadings between groups, or there's a difference in item means between groups. In order to sort of bend over backwards as much as possible and let other explanations possibly account for it, in this case we can't estimate a factor variance difference as well, but we can allow for factor mean differences between groups, so we fit these 3 and this parameter free. And conversely over here, with mean differences between groups, we can allow for a factor variance difference between groups. So I take those 6 models and throw 50,000 people's worth of data at it on 13 items, fit it by full information maximum likelihood, doing 13-dimensional numerical integration to compute the likelihood for each of the distinct observations in the sample, and because age is continuous, they become distinct quite easily. So comparing between these 6 models and looking at the data for marijuana use, here we see what looks like really pretty substantial age and sex effects on factor means and factor variances. That compared to this model with no effect of the covariates, we're coming up with anything between 35 and 500 some chi-square units in this test. Next we look at the effects down here, and these are compared to the model that says we've got age and sex effects on factor mean or variance up here, but we're going to compare that here, and this is evidence sticking out like a sore thumb saying we've got some measurement non-invariance. It says that age and sex effects on the factor loadings is giving us a chi-square of around about 92 on some 14 degrees of freedom, comparing between this model with this. And so something is a bit screwy with the measurement. This is also true of looking at the item means. So it's not just the factor loadings are screwy, the item means are also screwy, i.e. they are separately sensitive and differentially sensitive to group differences. So all of the group differences that we're looking at are going to be discolored by the item level differences. So aggregating into a score is not going to work as we would hope, be it a sum score of the items or a factor score of the items. Those are potentially broken. And the same kind of deal is true of the alcohol data. Again, pretty substantial likelihood ratio difference chi-squares. Rather less so for cocaine. Of course, the statistical power for the study with respect to cocaine symptoms is considerably less, because a much smaller proportion of people in the population use cocaine than alcohol or marijuana. And that's even more true for opioids. Although opioids have actually been making quite a comeback in terms of substance popularity, partly because of oxycodone being used for surgeries and other pain control. People started using oxycodone, and then they find out that heroin is about 1/10 the price of illegal oxycodone on the street, and so they start using heroin instead. Okay, so we've got this measurement non-invariance situation here. What are we going to do about it? And one of the things that you can do is, just like in standard MANOVA, if you like, that the overall test, the multivariate test is significant, so we're now justified to go and start having a look at the individual tests. Let's see, are there some of the symptoms of abuse or dependence that are non-invariant, the ones that are effectively the tails wagging the multivariate dog. So I wrote another little function for this. In addition, what it does is take one of the models that you've fitted. Having constructed that model with a function, I know where the bits are, and I can tell it to say, okay, let's look at measurement non-invariance for these particular factor loadings, or these particular item means, or these particular item variances. So on a single item by item, symptom by symptom basis, what's the evidence for non-invariance for each of them? And here would be an example use. Forget the coding exercise. And then, because that ends up being a lot of information, right? You could have measurement invariance with respect to sex, you could have it with respect to age, and you could have it at the item mean level or the factor loading level. In principle, you could have it at the item variance level, but these items were binary, so you have to take your pick, either item means or item variances, and I picked means. So then I drew a heat map plot. The wonder of having this stuff in R is that it's just a few more lines of code to spray the results of fitting and models like that into a heat map and say, haha, tolerance here looks particularly bad, a huge signal for invariance with respect to age. And that makes a certain amount of sense, especially when you think of alcohol and perhaps over a career of heavily drinking at conferences and workshops and things, your tolerance increases over time, and that tolerance may not be such a great measure of overall enjoyment of alcohol or tendency to get into problems with it, depending on how old you are. Similarly, some of the physical problems, that seems to be measurement non-invariant with respect to sex, and doing alcohol in dangerous situations. So you can again imagine, and the direction of effect is such that males tend to drink and drive quite a lot more than females, and probably that's why this particular item looks measurement non-invariant with respect to sex, something probably associated with externalizing disorders, which we men seem to find so much fun. So, having a look at marijuana, roughly the same kind of pattern here, getting into trouble with the law, a bit more of a problem for men than for women, perhaps. And with respect to age, not nearly as striking. The patterns of use of marijuana over the lifetime are rather different than for alcohol, that they tend to peak in the early 20s, maybe third or fourth year at college, and then drop off. That many more people who start using marijuana actually quit using marijuana than continue and go on to have a problem, at least up till recent legalization effects that may change that considerably. So we can look at the same thing as cocaine, but I think you get the same the principle, and there's a lot less information on the cocaine and opioids than there was for alcohol or marijuana. So what do I do, right? So I know that the scales aren't functioning as I would like. Is there something that I can do? And this is really a proposal and a conjecture, and I haven't sort of run through dozens of simulation studies, and I know there are a few potential issues with it, especially with respect to missing data. But what I want to do is use the moderated paths in order to compute people's factor scores. I want to be able to plug in whether they're male or female, whether they're young, what age they are, and plug that into the structural equation model that I'm going to use to compute maximum likelihood-based factor scores. So each person potentially is getting a different structural equation model that is used to compute their factor score. Ryan Estabrook and I worked on some comparison of factor score estimation, and like for most things, maximum likelihood's pretty cool as long as you can afford the computing time for it. So the general approach is use the old function that we had previously to fit the model, get the parameter estimates, sort out all the non-invariance effects, and then the conditional likelihood approach is it's just simple conditional probability, basically, saying that we're going to take the likelihood of the factor score, that is the density of the normal distribution at the top of the path diagram, multiply that by the likelihood of the item response data, the set of items, given the factor score. Conditional on the factor score, in a single factor model, the items are independent, so this makes computation quite a bit easier. Rinse and repeat this process for all subjects, and then we're going to compare the scores before and after correction. And so here's the factor score for alcohol with no correction in the model, and here's their scores after correction, effectively by including all of those items that look like they have significant factor loading or item mean effects. And what that sort of means is if you're just to pluck out one person and say, okay, how does this person compare to the rest of the sample? It's saying that this swath of persons down here are sort of effectively misclassified. They're thought to have higher scores on alcohol abuse and dependence than this individual. Conversely, these ones are thought to have lower scores, and that ordering gets revised by the change in scoring. So there are different sort of endpoints really in this work, one of which is getting to a point of giving people individual scores, and that's useful for certain purposes, be it charging insurance companies or getting people into substance use programs or whatever. But to answer research questions, by and large, you don't necessarily want to do that. In fact, if you can, never scoring, just directly analyzing all the item data that you collected in the first place, never aggregating into a score, doing everything in the model, statistically, that's as kosher as it gets, right? The two-step analyses are always more problematic, but often they're a good deal more pragmatic. So doing this kind of modeling with genome sequence data, for example, and saying, does this SNP have an effect on the, and so on, could be particularly challenging to your ability to publish before you die. So, and it's publish or die, preferably not sort of both. And there's an order effect there too, right? So this is best in principle, but sometimes it's just not practical, and that's why we would need scores at all. So another perspective on the results of factor models is to just translate them into item response theory terminology, to think about what's the probability of responding to a particular item given where you are on the underlying trait liability. So here's what we think of as the population distribution of the latent factor, and here's perhaps your chance of saying yes to a certain substance, conditional on where you are in the distribution. So you want to be at sort of 50-50 point here to say, okay, I've now got a 50-50 chance of responding. And those factor loadings really translate into how steep this curve is, and the item means translate into where that 50-50 mark appears on the underlying distribution. Okay. Now, you can then say, look at different symptoms, and these are symptoms for the Fagerstrom test for nicotine dependence, a number of items testing essentially how much you like smoking and how addicted you are to it. And some of them have smaller factor loadings, and some of them have larger ones. What's more tricky is to compare across substances. And it's possible to compare across substances as long as you know the relationship between initiation of that substance and going on to having different symptoms. That correlation is pretty much key, because it doesn't just affect the chance of responding to these symptoms, but you really have to correct for the base rates of the symptoms in the population, and correct for the base rate of using the substance, but also the correlation between the two. And one way to do this through ML is actually to mark all of the people who didn't initiate as missing on their substance abuse and dependence items. That's missing data. A lot of people will score that as 0 in terms of symptoms, but scoring them as missing is actually getting at a different question, an underlying propensity. And you can think of times when it's just not realistic. You know, if I really like cocaine and I really like marijuana, but I've never tried heroin yet, scoring me at 0 for heroin addiction propensity isn't the right thing to do. I might have 0 active addiction, but I don't have 0 addiction propensity. So what we were trying to do here was to get hold of the symptoms. This particular symptom of using it more or longer than you planned showed up as being especially prevalent for cocaine. That cocaine being the leftmost curve means that the largest proportion of the population who actually use cocaine are likely to find this. Much larger proportion will get this symptom than, say, for marijuana, which shows up near the right-hand end of the distribution, as perhaps you might think. Okay, just in passing, a note that modeling the latent trait as being normally distributed is something we routinely do, but we don't have to. It is possible to separate the distribution of the latent trait from the distributions of the items that go up to make it. Often you will see a sort of horrifying pile-up of zeros, zero inflation that people will say have no symptoms on the scale. That doesn't mean to say that the latent trait is non-normally distributed. It may or may not be. And there are methods. There's one that a brilliant grad student, Eric Schmidt, and I published. He's not the Eric Schmidt in charge of Google. This is another one. That provides a test for normality of the latent trait. But that I'm not going to talk about, but it does provide a bit of a segue into thinking about other kinds of models of liability to addiction. And these are models that some of them have been around a long time. So the factor model is a bit over 100 years old now. The latent class model, a bit over 50, kind of like myself. And then there's a more recent factor mixture model that sort of blends the best of both worlds, but is as yet not terribly widely applied. And I also want to talk about a third flavor of model, which is one that doesn't have any latent variables, which you might count as heresy with my background in structural equation modeling. But I think it's entirely plausible, and probably the world works in all three of these ways to different degrees with different collections of symptoms. So the latent class model has this rather curious assumption that within a class all the symptoms are conditionally independent. There's some probability of being in one class or being in another, but whichever class you're in, all of the items are independent. This was sort of somewhat extreme, and the factor mixture model is a bit prettier than that, and it says, actually, I have got a mixture distribution in the population. There are two flavors of people out there, but within each group, there's still a factor model operational. And latent growth curve mixture model is an example of a factor mixture model. We applied this to data on alcohol symptoms, and came up by basically spamming the data, if you like, with models with different numbers of classes, different numbers of factors, different numbers of classes and factors, and then trying to figure out which one fit the data best. And we came up with a one-factor, three-class model, so a factor mixture model with different proportions in the classes for males and females. And these are trying to cut down withdrawal symptoms, tolerance, and so forth symptoms, differentially plotted for the different classes and the different sexes. So that mutualism model that I mentioned, this, what I would call an NLV, no latent variable, we've got just regressions drawn from every variable to every other. Those of you with any familiarity with these, with regression, for example, will realize that I can't identify a unique set of parameters for this model for all of the paths in the model. We start off thinking that each of these has their own variance, but they may have knock-on effects. It's an entirely plausible kind of thing. You can imagine, as say in the case of depression, of sleeplessness, perhaps causing a lot of the other symptoms, messing with your appetite, making you weepy, making you irritable, and so forth. But you wouldn't be able to get a hold of that if you just measured unrelated persons in a sample on those 5 variables on one occasion. You can get something of a hold of it in, if you like, a variant of cross-lag panel analysis by measuring on multiple occasions. But one way that I've been playing with is looking at it in twin data. So you'd let these variables correlate between twins, but exactly the same values of these paths are there for both twins. And that model is identified. So it gives you a chance of saying, the strength of the regression from x3 to x2 is greater than vice versa, for example. And in reality, probably there are both factors and this kind of mutualism thing going on for a number of the symptoms and collections of disorders and diagnoses that we measure. And figuring out what combinations of, I'll take 3 factors and 12 regression paths or whatever, which ones are identified. Well, if anyone's looking for a postdoc, I'd be really happy for them to come and help me figure that out. Okay, so next topic. Not doing too badly on time, but not brilliantly either. Growth curve models, growth curve mixture models, and some regime switching models to think about, okay, what do I do with developmental data? And one popular thing to do with developmental data is to fit the so-called linear growth curve model that just says there's variation in level and there's variation in slope, but the slopes are all linear, and if you follow that series out, it means that you've got infinite variance virtually, and by and large I would say that just a linear growth curve model is silly, and acknowledge John Cleese for the Ministry of Silly Walks demonstrating variation in slope. So, one solution, of course, is to add a polynomial component, a quadratic component, and something that Jack McArdle and I did years ago was to implement different types of growth curves. This was based on seminal work by Michael Brown in the early 80s, looking at particular functional forms, logistic growth curves, exponential, Gompertz, anything from the Richards family, if you like. And you can also work that kind of thing into this type of model. It's essentially a matter of getting the partial derivatives of the components of the growth curves plugged into parameters, and that if you have software that lets you use algebras to specify what these things are other than just constants, that becomes possible too. But we'll stick with the quadratic growth curve model here for a bit and say, again, just like the latent class case, anybody in the population might be belonging to one class or the other with some probability p or 1 minus p. And then apply it perhaps to some substance use data. And it was my great friend and colleague Connor Dolan who was, I think, one of the first to say, well, perhaps people aren't just stuck in one class all their lives with no chance of changing whatsoever. So that sort of taking a bit of a swipe at the deterministic nature, even though it's just probabilistic, whichever trajectory on, you're stuck. That's it, that's what you do. And for substance use data, that's not necessarily the best thing, right? Because you may have experienced this yourself, but you might go through a period of lots of substance use and then be on the wagon for either the week or for a few years, but then start again. And that type of pattern of starting and stopping use is not well captured by even a latent growth curve mixture model. So the idea is that you can flip from one component or regime to another. And applying this to national longitudinal survey data, looking at 700 odd people at various different ages. They started off at 13 or 14 and were examined annually after that. And the question here, this is so key, and there's a lesson in survey design hidden in this question that I hope will come out later. But the question is, in the past 30 days, on days you drank, how much did you drink? And this was made into an ordinal scale and analysed with a growth curve mixture model that permitted some probability of transition from one class to another. So a Markov process, essentially. You can keep those probabilities the same between adjacent ages, or you could let them be different, or you can say that I've got a second order Markov, that it's not just what happened to me where I was last time, but maybe the time before also influences my probability of changing from one class to another. But the regime switching model fitted really dramatically better than the growth curve mixture model. I'd forgotten about the size of the difference in fit, but it was hundreds of chi-squared units, a couple of hundred. This was what the growth curve mixture model plot looked like, and the regime switching plot had not much in the way of slope. And perhaps that's because when people start drinking, that there isn't, when you only measure them every year or two, there isn't that much time to sort of capture the growth component. They adopt a particular level and they stay fairly static at that. But there are three different flavors of how much people drink, probably hardly at all, weekends, and most days, something like that. Here were the regime switching probabilities, and this is with the model where they're equated from one age to the next. I won't, in the interest of time, show the model with different probabilities, but there's a good deal of stability at this age and rather less as they develop as we measure at later ages. Okay, so in this case we had a three-class model. We have 4 different occasions, and at any occasion you could be in any class. This is 3 to the power 4, which means there were 81 different components of the model. This is a mixture distribution model with 81 possible individual trajectories. And again, I wanted to be able to get back to, okay, suppose I've got somebody in the clinic and they're wanting to say, what's likely to happen to this person if I'm trying to make a prediction, or at least to try and interpret individual level data. So I used a different kind of plotting, just in sort of rainbow colors, where red at the bottom is nothing but light, and then violet is where you're heavy all the way through. And so you get something looking like this. Here's the red stuff at the bottom, the violet at the top, and there are lots of different lines here, right? So 81 by the time you add them all up at the rightmost occasion. That's, and I use thickness to say, okay, what's the probability of being in these different classes? So at the end of model estimation, we get prior probabilities of these different components, and there's more of the folks on each of the particular levels than there is typically changing between them. Take that back and look at an individual vector of data and say, okay, what's the likelihood for under this model, what's the likelihood under this, and so forth, and look at those 81 different likelihoods and add them up, divide each one, and say, I'll make this line as thick as the relative likelihood that they're in the different trajectories. And so for this individual, and the amount they reported drinking is shown in the blue dots, and this, lo and behold, it was something of a relief to see it come out like this. This seems to be the most likely trajectory for that person. This person's a little bit strange. Looks like they were getting pretty smashed when they were 13, 14, drinking between moderate and heavy amount, then more or less gave up, and then started to reuptake the habit, but to a lower level by the time they were 17 or 18. Wonder what's going on with that. And so you can plot these trajectories, you know, and I did for all 737 people in the sample. Here's one where we didn't have any data on the first occasion, and it's saying they won't have much of a problem later on. And here's one with prediction, and you might be worried like, oh, is this person going to go up here and end up a heavy drinker? But no, the model is saying, I think actually they're going to stay a moderate drinker in this case. The rider to all of this, and if we go back to that question, I think that what happened is that the younger kids answering this question didn't sort of let it sink in all its glory, and they were answering how much they drank in the month instead of how much they drank on a particular day. Because otherwise some of these kids, in fact, looked like they were drinking a case of beer every day, and that was definitely suspicious. So we did take those really extreme cases out of the data set and got more or less the same answers. But talk about a lesson in survey design, and this is a survey that goes out to tens of thousands of people. Time-wise, now I could talk a little bit about the twin stuff and direction of causation, but the hour is up, so it's sort of your call.
Yeah, I'd like to hear this.
You want to hear this? Okay, yeah. I guess people should feel free to leave, and I'll feel free not to feel offended if you do. Okay, so the twin study gets a certain amount of credit actually in general epidemiology with the idea of a co-twin control study. So you have say a pair of identical twins, right? You know they've been brought up in the same home and same sort of socioeconomic status, and their genotype is identical. So that's like a large chunk of things that could be making people different from each other that is elegantly controlled if, say, one of them starts smoking. So one of them starts smoking, and then you examine say the brains of these discordant MZ pairs, and you say, aha, the one who's been smoking, their brain is shrinking. From that we would infer that it looks like a direct causal effect of smoking, and not just that people who like to smoke have a genetic tendency to have their brains shrink as well. Yeah, so there's the principle of the discordant twin design. But it turns out that that's okay for sort of expensive secondary protocols, but often we have lots of epidemiological data, twins, MZ and DZ and so on, and they're measured over time, and you can extrapolate from all of the data rather than just focusing on the ones that you've declared discordant, or just focusing on the differences between those twins at the original time point. I would argue that both of those methods are the focusing on the differences or the discordant pairs can be really wasteful of data. If you've got all that data, throw it in the model, get the likelihood of everything that you've observed, and work with that, rather than focusing. That's different experimental design, essentially. So how does causal inference work in the general twin design? And in fact, you just need pairs of relatives. You don't have to scout the country for twins, and this is one of the reasons that we're trying to tie together genomic studies with twin studies more tightly, so that we can do this kind of thing. But even between myself and everyone in the room, you compare all of our genotypes, we all have this tiny bit, sort of plus 0.05, minus 0.05 correlation between our genotypes. We could use that to draw the regression line of similarity, and we could do that separately for different phenotypes. So what we're saying here is that if you have this putative endophenotype, say a measure of the brain, and you think it influences the substance use disorder as an outcome, and the endophenotype correlates between the twins, and there's some other level of correlation between substance use disorder. If you draw the regression this way round, it makes different predictions about the across-twin, across-trait correlation. Here, it's going to be REP, the correlation between the endophenotypes, multiplied by the correlation, essentially, the regression between these two things. So you've got REP times B. But if causation was working in the other direction, that the taking drugs was frying your brain, then you would see a different predicted cross-twin, cross-trait correlation. It would be more consistent with this size of correlation than this. So as long as these two correlations differ, you have some power to resolve the direction of the regression. And you can, in fact, fit the model with regression in both directions. And so this is the bivariate flavor of the mutualism model I described earlier. And kind of for shits and giggles, really, I just applied it to this data on brains and smoking. I mean, not entirely frivolously, but knowing that the statistical power to resolve was not likely to be all that brilliant. But just to take a look under the hood, so we've got a couple of hundred twin pairs. They've had MRI scans, the 1.5T, because this was done quite a while ago, and you from that can extract volumetric measures of different regions of interest in the brain. So somebody else did that for me. Wonderful, great. And I got to play with the volume data in this particular analysis. Although there's a good argument for not using cortical volumes, but for using cortical thicknesses and cortical surface areas separately, as we subsequently figured out that these look like they haven't got anything to do with each other genetically. That limitation notwithstanding, here were some regions that showed up as being significantly associated between amount of smoking. And this is in a sample of twins unfortunate enough to be born in the era when they were drafted to Vietnam. There were some 4,000 pairs of veteran twins from the conflict that were ascertained by folks at the VA in Boston that formed this study. Ming Swong and Mike Lyons were the real creators of the project. So I fit the direction of causation model and say, okay, we've got a measure of pack years, and like how much cumulatively essentially have these folks, who are all pretty homogeneous in age, within 10 years of each other, has each person smoked, and fit that direction of causation model to the data. And The direction of effect is sort of smoking causes your brain volume at the top of the figure, then below this line the weight of evidence is in the other direction. It's noisy and it doesn't ever reach a particularly high level of significance, sort of a chi-squared of a couple or so. But at least it's sort of trending in the direction, and these are for 35 different regions, but the last two here are across the brain as a whole. So that's left hemisphere and right hemisphere over here, and they're trending in the direction of the weight of evidence. Looks a bit more like smoking reduces your cortical volume than the reverse. This isn't anything to sort of write home to the Surgeon General about, but might be the beginning of perhaps findings that would say, hey, if you're worried about your brain, then maybe you shouldn't smoke so many cigarettes. In other work, we've kind of tried to map the brain using bivariate genetic models and saying, okay, have I got regions of high genetic correlation within a region and less genetic correlation between regions, by basically fitting tens of thousands of bivariate genetic models to the data, and then cluster analyzing an enormous genetic correlation matrix. And we came up with this, which actually bore a bit of similarity without too much stretching of the imagination to regions of gene expression on the mouse brain, which so my excellent colleagues in UCSD were able to publish this in Science a few years ago. But it would be fun to use these predefined regions and say, when I'm doing genetic correlations between, say, substance use or sensitivity to trauma, to focus on regions defined genetically rather than morphologically or functionally. So there's a bunch of future directions, and hopefully some return to the acknowledgement slide, which to my surprise I've actually had time to get to. So thank you.
Questions?
Yes.
So how did the twin data get into the brain study? I mean, were you looking at the invariances across the twins, or in that brain volume?
In this one, or
These are the twin data, right?
yeah, this is out of the twin data. So
Okay. So how are you modeling? How
Right.
are you modeling the two twins to get these maps?
right. I could probably draw a diagram for you easier, but the essence is, in the classical twin study, we're saying that the identical twins are more similar than the fraternal twins because their genotypes are more similar. You can apply the same approach not just to variation within a trait, but to covariation between traits. So to the extent that the across variable correlation is higher in the MZs than the DZs, it's implicating genetic factors in the covariance. And so we then did that analysis. It's a fairly simple 4-variable analysis, just 2 traits, 2 twins, and rinse and repeat that analysis between 5,000 different points on each hemisphere in order to build up a 5,000 by 5,000 genetic correlation matrix. It turns out that the genetic correlations from left to right are extremely high. That and you know, that's sort of well, it's just some sort of regional thing, topographical thing. Not so much. Not when these genetic correlations are really high, but these aren't. And so drawing drawing then a map, basically summarizing that enormous genetic correlation matrix by cluster analysis to say, okay, if I'm in this region, the genetic correlation between the voxels there is higher than it is across regions. But it's, you know, approximate, and heaven knows it could certainly use some replication.
So it's also approximating to a sort of universal brain.
Well, yes, and that is still sort of remains an issue, but in the mapping of the data that's collected by the MRI machine, they're sort of projected onto, if you like, a spherical version of the brain. And a constant concern that I have, that some of the reasons that we get high heritabilities is that maybe simply morphologically, the MZ twin brains look much more similar, and you can sort of see pictures of brains in jars, and the MZ twin brains look really similar and the DZ ones don't. And what I'm hoping to do, should Eric Schmidt is in particular, who being MD Ph.D. went on to become a radiologist, and so he's been doing nothing but brain scans for like the last 5 years. If he comes back and he's got much better understanding of the mathematical mapping between the brain and the enlarged image, and being able to measure the error of that, then perhaps we can control for what is correlated error of measurement in the twin data. Things I don't sleep too badly, but sort of things that can make you worry about at night. Yes?
Just to understand these pictures, the colors that are the same are the correlated areas, but
Yep.
each of the different colors are uncorrelated with others.
Yes, not entirely uncorrelated, but definitely lower correlations, yeah.
And so you're finding on the surface more clusters and then kind of internally fewer?
It's all
3, 4, 8, 11, 12.
right. It's
Or maybe it's just a different slice.
yeah. So these are different views of a brain, of the cortex. But
And the lighter and darker don't really mean anything. It's different.
no, that's not. Unlike the rest of my talk, it has no information. Yes.
I'm curious to hear your response. So I get reactions from our biological colleagues when I bring up latent variables or latent classes. They'll say, well, if you don't directly observe these things, why are you trying to build biological connections with biology? So I've had a hard time getting them to see the value in latent approaches to phenotype data. What's your response to that?
The usual one is, I'm very jealous of you being able to measure things completely without error. If you think that you might be able to measure things without error, good on you, but admitting of the possibility that there's error of measurement is really I think the starting point for latent variable modeling. And being able to estimate that, and perhaps even being able to pull it apart a bit further. So the factor model for twins will have the factors for the twins correlated, but we'll let the residual components of variance correlate as well. If that's purely error, you're not going to see any twin correlation for it. Sometimes you do, and sometimes you don't. And the other part is that controlling for any possible error is at times analytically very important. So in the direction of causation modeling case, for example, there will tend to be bias that the more reliable measure will look like it's causing the less reliable one. It's hard to get an unreliable variable to cause anything. And latent variable modeling is a very direct way of passing reliably measured components of variance out of unreliably measured ones, and then running the direction of causation modeling at the latent variable level is less likely to be led astray by error of measurement. It's not necessarily an argument I always win, of course.
Anybody else? Questions? All right. Well, thank you.
Thank you.